Welcome to the Deep Dive. This is where we sift through some of the most complex, really cutting-edge research to give you the essential knowledge and those aha moments that actually matter. Today we're following a story that doesn't just span technologies. It really spans the very philosophy of how scientific research gets done, and that's accessibility.
That's absolutely right. And this deep dive is, I think, a bit unique because we're not just looking at a shiny new piece of AI. We are actually tracking the intellectual blueprint of an innovation.
We're tracing its roots.
We're tracing its roots, exactly. Back to this foundational work done years earlier, work that was focused on one thing: making specialized knowledge usable by everyone, not just, the programmers and the bioinformaticians.
Okay, so our mission today is to unpack two interconnected scientific works. The first one, it dates back to 2021 and sort of established the gold standard for creating a user-friendly scientific workflow, specifically in oncology. It's titled Galaxy and Mean Stacked to Create a User-Friendly Workflow for the Rational Optimization of Cancer Chemotherapy. It basically proved you could tame this incredibly complex bioinformatics.
And then the second piece is a very recent preprint that takes that proof of concept and just catapults it into the LLM age. It's called Conversational No-Code and Multi-Agentic Disease Module Identification and Drug Repurposing Prediction with Chatbots.
Okay.
But the fascinating part, and this is really the core of our whole discussion today, is recognizing why the authors of Chatbots cited that older paper. They used it strategically to justify their own existence.
So this is a story about a huge transition then. We're moving from complex systems biology that requires, massive programming expertise to something that's approachable, conversational. We're going from highly specific Perl and Python scripts running on a server somewhere to just asking an AI agent a complex biomedical question in plain English.
And the central problem, the thing that really connects these two very distinct projects, is this persistent and honestly debilitating issue in science.
Which is what?
Systems biology and drug development. They rely on these immensely powerful computational tools. I mean, think about network analysis, which maps how all the genes and proteins interact in a disease.
Right. Incredibly powerful stuff.
But historically, to access those tools, you've needed a substantial degree of bioinformatics and programming expertise.
It just creates this massive chasm. The person with the deepest clinical insight, the oncologist, the translational scientist, they're usually the furthest away from the skills needed to actually run the tool.
Exactly.
You end up needing an expert intermediary for every single question you want to ask the data.
And that bottleneck, it just slows discovery to a crawl. So our goal today is to examine how that 2021 paper, it set up the blueprint, both architecturally and philosophically, for making that complexity accessible.
And then how Chatbots, years later, completely realized that vision.
Exactly. By creating a multi-agent system that I think finally fulfills the promise of truly democratized biomedical computing.
Okay, let's unpack the origins of this then. Let's start with that 2021 work on cancer chemotherapy. What was the driving scientific goal behind developing the Galaxy and Mean Stack workflow in the first place?
The motivation was, at its heart, translational and profoundly important. It was about personalized oncology.
Okay.
We're talking about moving really decisively beyond this outdated idea of one-size-fits-all chemotherapy. We know, especially in cancers like breast cancer, that heterogeneity is just rampant.
Right. Two patients, same diagnosis on paper, but their tumors are behaving completely differently on a molecular level.
Precisely. So they wanted to tailor treatment to rationally optimize the drug choice based on the unique genetic signature of an individual patient's tumor.
So you're basing the therapy on their specific molecular phenotyping.
Yes. It moves the decision-making process from, traditional clinical staging to a data-driven understanding of the malignant mechanisms at play in that specific person.
And the key to unlocking that disease mechanism wasn't just looking at, say, which genes are highly active, but how those highly active genes were all linked together. This is where network science comes in, right?
That is the core insight. They weren't looking at gene overexpression in isolation. They developed a strategy that was all about identifying what they called connection hubs within protein interaction networks, or PPI subnetworks.
So they would start by just identifying genes that were way more active in the tumor tissue compared to the healthy tissue right next to it.
Exactly. Compared to the adjacent non-malignant tissue, the stroma.
So if a gene is way more active in the tumor, it's a potential target. But I guess the network analysis tells you which of those targets are the most efficient to hit.
That's it. That's the whole idea. The underlying network theory confirms that you get the most systemic disruption by inhibiting proteins with high connectivity, the hubs.
Right.
If you think of the tumor signaling network as like a massive interconnected city,
Yeah.
you don't want to cut a tiny little side street. You want to take out the main freeway interchange.
Because that causes a cascade.
A cascading failure across the whole malignant signaling network. It maximizes the therapeutic effect.
That makes perfect sense intuitively. But something like network analysis feels like it would be incredibly sensitive to the quality of your input data, the parameters you pick.
Oh, absolutely.
So how did the researchers objectively validate that their entire methodology, I mean, from the raw data processing all the way to identifying these hubs, how did they prove that it actually produced results that mirrored real-world clinical outcomes? That seems like a huge scientific hurdle.
It is. And this is where the work, I think, truly separates itself from less rigorous approaches. They didn't just assume it worked. They used a concept from information theory.
Okay.
Shannon entropy. They used that as their objective validation metric.
Shannon entropy, I usually hear that term in coding or signal processing. How does that translate to measuring the aggressiveness of a cancer network?
Yeah, that's a great question. So think of Shannon entropy as just a mathematical measure of uncertainty or complexity within a system.
Okay.
In a PPI subnetwork, it quantifies how many different connectivity values there are. So how many different ways proteins are connected and how evenly all those connections are distributed. A simple, really organized or redundant network has low entropy.
But a highly complex, decentralized, constantly adapting network
Was an aggressive tumor.
has high entropy.
It has high entropy. The formula basically calculates the probability of finding a protein with a specific number of connections, and it sums up all that uncertainty.
So the hypothesis then is high entropy equals high complexity, which should directly correlate with a more aggressive, harder to treat cancer.
That's exactly it. So the validation process involved calculating the Shannon entropy, or HA, for these subnetworks that they derived from RNA sequencing data, and they did this across 9 different cancer types.
And then compared that entropy value directly to patient outcomes.
Directly to the patient's 5-year overall survival data. And the key, really groundbreaking finding was this precise, quantifiable negative correlation between the subnetwork entropy and the patient's survival.
So higher complexity, higher entropy.
Lower patient survival rates.
Wow. That correlation is the linchpin then. It's the objective criterion, the hard evidence that their computational model of the tumor's molecular landscape actually reflects the real-world clinical behavior of the cancer.
They found a quantifiable molecular marker for aggressiveness. And this is where we have to, dive into the technical depth a little bit, because that's what validates the whole approach. This negative correlation didn't just validate their hypothesis.
Okay.
It became the mechanism for validating the entire upstream data processing pipeline.
Because that raw RNA-seq data is messy, right?
Yeah.
You have to normalize it to account for all sorts of variables before you can even compare gene expression levels.
It's incredibly messy. You have to account for sequencing depth, gene length, all of it. They had to be sure they chose the right normalization method, one that accurately captured the tumor's true complexity. Otherwise, the whole model is just garbage in, garbage out.
So they tested a bunch of different ways to clean up the data.
Absolutely. They rigorously tested several normalization methods, and they found that the optimal combination, the one that gave them the highest magnitude of that negative correlation coefficient, was something called RPK-MUPPER combined with a log2 transformation.
Okay, RPK-MUPPER plus log2. That sounds like pure jargon to most of us, but you're saying it's the difference between a validated clinical tool and just guesswork.
It is. Let's break it down real quick. RPKM stands for reads per kilobase per million mapped reads. It's a standard way to normalize RNA-seq data.
Right.
RPK-MUPPER is just an enhancement that stabilizes the quantification, makes it more robust against outliers. And then by adding the log2 transformation, you compress the huge dynamic range of gene expression data.
Because some genes might be expressed, hundreds of times more than others?
Thousands, even. And the log2 transformation brings those massive differences onto a scale where the network algorithms can actually treat them more evenly and meaningfully.
So they were essentially tuning their mathematical lens. They ran the data through all these different normalization filters, calculated the entropy of the network each time.
Yes.
And then looked for the result that most tightly aligned with the actual patient survival data.
Precisely. And the RPK-MUPPER plus log2 configuration was the clear winner. It produced subnetwork sizes that were biologically realistic representing only about 2 to 5% of the human proteome, which is what you'd expect for a disease-specific module.
And most importantly,
it yielded a correlation coefficient of R equals minus 0.91.
wait, an R value of negative 0.91 for a biological system, correlating a computational metric to real-world patient survival.
I know.
That magnitude of correlation it's extremely rare. I mean, you almost never see that in translational medicine. It signals an almost deterministic relationship.
It really speaks volumes about the rigor of the approach. The methodology's success wasn't defined by, whether the code ran without an error. It was defined by finding the data processing steps that maximized the objective mathematical correlation between their complexity measure entropy and the real-world measure of cancer regression 5-year survival.
And that rigorously validated pipeline became the basis for identifying the most connected proteins, which they then proposed as the optimal drug targets for that individual patient.
That's it.
Okay, so they have this highly validated, robust analysis process. It involved multiple complex steps written in Perl and Python. Now comes the second challenge, and arguably the more critical one that sets the stage for our AI story.
Right.
Making that complexity usable by healthcare professionals who do not and really should not have to be programmers. This is the accessibility blueprint.
This is the pivot. The focus shifts from can we do this complex math to how do we deliver this complex math to the bedside.
So they took the core analysis algorithms,
Yeah.
these steps like count connections, critical value calculation,
ETP, PTTCS, all of them.
and they encapsulated them within the scientific workflow platform Galaxy.
Exactly. Galaxy is essentially a laboratory workflow management system. It lets you plug together different pieces of code, a Perl script here, a Python script there, and run them sequentially without the user ever having to touch a command line.
It automates the whole thing. But even running a Galaxy workflow can still be a bit intimidating, right? It often requires a specialized interface.
It does. And to achieve true accessibility for clinicians, they went a step further. They built a dedicated, secure front end using the mean stack.
Mean, for our listeners, that's a full-stack JavaScript environment, MongoDB, Express.js, Angular, and Node.js. Why was that specific tech stack so strategic for this clinical application?
The choice of Angular for the front end was absolutely critical. Angular is a really powerful framework, and it allowed them to build a single-page application, or SPA, where complex data validation and even some heavy calculations could happen client-side.
Right in the user's browser.
Exactly, and that significantly reduces the load on the server, which ensures it's really responsive. That's essential in a clinical setting where time is critical.
And it also ensures data integrity, I'm guessing.
Yes, very much so. They used Angular's features, like reactive forms, which enforces these rigorous validation rules before any data gets submitted to the server. So if a clinician uploads a file that's, incorrectly formatted or it's missing a key identifier, the system flags it immediately.
Smart.
And furthermore, the use of Angular Flex layout was crucial for implementing what's called responsive web design, or RWD.
So that's the ultimate practical requirement. The goal was never just a desktop website. It was meant to be a portable diagnosis portal.
Precisely. They engineered the design so it would function seamlessly on small and mobile devices. They even explicitly cite devices like the Galaxy S5 and the iPad in the paper.
So a doctor could literally pull out a tablet on their rounds, upload a patient's data, and get back this sophisticated analysis.
A healthcare professional needed to be able to execute this sophisticated cancer diagnosis, complete with personalized target identification, without ever needing bioinformatics knowledge, programming skills, or even a specialized computer terminal. They created a complete, validated, end-to-end theranostics solution.
That's phenomenal for 2021. They proved that network science, validated by deep biomathematics like Shannon entropy, could be packaged and delivered to the point of care through a simple, secure web app.
And this foundation, this demonstrable proof that the complexity can be externalized and the user interface can be simplified, that is the intellectual bedrock upon which the next generation of tools like Chatbots would build. They didn't just solve a cancer problem, they solved an accessibility problem, and that set a precedent.
Okay, so let's fast forward. We go from that personalized oncology optimization to the broader, I'd argue more complex, field of drug repurposing. The problem of accessibility didn't go away.
No, it just scaled up dramatically. While the Pyers paper focused on optimizing chemotherapy based on one patient's unique profile, the demand for accessible tools just exploded in drug repurposing.
And that task is just constrained by so much more data.
Right.
It's a massive, fragmented.
Oh, it's a nightmare. You have pharmacology databases, genomics, clinical trial literature, huge knowledge graphs. And sure, specialized tools exist to navigate this, like the ones in the Nedrex platform. But using them requires, navigating complex database schemas and command line interfaces.
It's the same old bottleneck. It's just now applying to hundreds of thousands of data points instead of a single patient's file.
Exactly. And this is why Chatbots is introduced as this transformative solution. It's the next logical step in this lineage of accessibility we've been talking about.
So what is Chatbots fundamentally?
It's a conversational multi-agent system, and it's designed to provide natural language access to these highly complex network-based drug repurposing analyses.
This really is the fulfillment of that original vision then. Instead of having to learn the data entry standards of a web form or the sequence of a workflow, the user just opens a chat window and asks the system, what are the most promising existing drugs for this specific disease mechanism?
That is the power. It's democratization through large language models. It empowers the clinical expert, the person who actually understands the medical nuance and needs the answer, to control the entire advanced bioinformatics process just using plain language.
That could accelerate hypothesis generation, therapeutic discovery, exponentially.
That's the promise.
But okay, let's be responsible here. When we talk about leveraging generalized AI, especially LLMs in a high-stakes field like biomedicine, we immediately hit the wall of hallucination.
A big one.
Right. A generalized LLM is notorious for sounding incredibly authoritative while just making things up. How does Chatbots manage that risk in a domain where an error could be catastrophic?
This is the fundamental engineering challenge they had to solve, and it informs the entire architectural design. Chatbots explicitly acknowledges and then counters the inherent unreliability of these generalized LLMs. It tames the beast by improving three major structural controls.
Okay, let's walk through those controls. What's the first one?
First is validated integration. Chatbots does not rely on the LLM's own internal static training data for biological facts or for running algorithms.
Okay.
It integrates directly with validated external data services and specialized bioinformatics tools, specifically the Nedrex knowledge graph and its computational methods. The LLM is forced to use the tools it's given.
So the LLM is more like an interpreter or a project manager, but the heavy computational and factual lifting is done by proven, specialized software.
Precisely. The second control is grounding. It uses retrieval-augmented generation MAGI and in-context learning. This means every assertion and every finding has to be grounded by retrieval from an external, validated biomedical knowledge base.
So the system doesn't just generate a plausible-sounding answer. It generates an answer that is actively verified against outside context.
Yes. It makes the LLM less of a creative author and more of a high-level manager and reporter who is required to provide receipts for every single action and conclusion.
Okay. And the third control?
The third, and I think the most innovative control, is the expandable multi-agent design. This is the core architectural innovation here.
So instead of one massive monolithic LLM trying to do everything.
Right. Instead of one LLM trying to handle knowledge graph querying and network analysis and statistical validation and literature review, all tasks it would likely struggle with Chatbots delegates. It gives these complex, deterministic tasks to a team of specialized autonomous agents.
It's like a system of checks and balances.
It is, and it mitigates bias and error dramatically. The specialization is key. It's like they created an AI research lab where every team member has a very highly defined specific job.
Let's dive into that architecture now then, because that's the technical roadmap for how you actually achieve this conversational no-code.
Okay, so to understand this, let's conceptualize the structure. The entire system is modular. It's built around an orchestra of these specialized agents, and they're all coordinated by one master controller.
And that master controller is the planning agent, sort of the CEO of this AI research team.
Exactly. The planning agent is the meta controller. Its primary role is orchestration and memorial management. The architecture follows an adapted chain of thought paradigm.
So traditional chain of thought is when one model sort of reasons out loud to get to an answer.
Right. But Chatbots delegates that reasoning. The planning agent takes a complex user query like find promising drugs for Alzheimer's by analyzing its disease module and the literature, and it breaks that down into a fixed sequence of subtasks.
And we know from the paper that it's a 6-step process, right?
Correct. N equals 6 for a full workflow. The planning agent assigns these subtasks iteratively to the specialized agents, it monitors their execution, and it maintains the overall state and memory of the whole interaction.
So it makes sure that the results from step 1, say, identifying the right genes, are correctly passed to step 2, running the network algorithm.
Precisely. And this modular approach, it maximizes efficiency, but crucially, it also allows the system designer to vet and validate each small step independently.
Okay, let's start with step 1, accessing the data. In network-based drug repurposing, that means querying the knowledge graph. That's handled by the knowledge graph agent.
Yes, and this agent's job is to translate fuzzy human questions into precise database language. And this is a massive leap in addressing the hallucination problem.
How so?
The KG agent's design is highly constrained. It doesn't generate data from thin air. It translates natural language questions into deterministic cipher queries.
Cipher, that's the query language for the Neo4j graph database, right?
Mhm.
So the agent has to know how to navigate all the nodes and edges in that graph structure.
Exactly. It has to map the user's intent to the fixed structure of the NEDREX knowledge graph. But here's the really critical innovation that prevents hallucination. The LLM-based agent doesn't just guess the entity names, like a gene name or a disorder name. If you ask about Huntington's disease, it doesn't rely on its own memory of that spelling.
So if I, slightly misspell a gene name, the LLM won't just generate a query that comes back with 0 results.
No, it won't. For any elements that require filtering, the KG agent uses an embedding-based similarity query against the knowledge graph itself.
What does that mean?
It looks for the top n nodes in the actual graph that are most semantically similar to what the user typed. It finds the validated entity ID first, and then it uses that verified ID to construct the deterministic cipher query against the known schema.
That's brilliant. It means the LLM is only generating the structure of the query, but the factual identifiers are pre-validated by checking against the source data itself.
It's like a librarian who verifies the ISBN before even looking at the shelf. It ensures the database query is robust and accurate, and it dramatically reduces errors.
Okay, so once the relevant genes or disease nodes are identified, the work moves to the heavy computational lifting. This is the NEDREX agent.
Yes, and this agent encapsulates the specialized network algorithms.
And which algorithms are we talking about here?
3 primary ones, all crucial for module detection and prioritization. The first is DIAMOND. That stands for Disease Module Detection. This is the mechanism for uncovering the hidden entire web of disease causation, not just the starting point.
How does DIAMOND work conceptually?
It takes the initial seed genes that the KG agent identified, the ones we already know are involved in the disorder, and it iteratively expands the network outwards. It identifies other proteins that are highly connected to those seeds in a way that's statistically significant.
So it builds this tightly clustered subnetwork, the disease module, which represents the collective mechanism of the disease inside the massive human interactome.
You got it. It identifies the functional neighborhood of the disease.
Okay, now that we have that neighborhood, how does this system figure out which drugs are most likely to disrupt it?
That's the role of trust rank and closeness centrality.
Trust rank. I've heard of that from Google and web spam.
The very same. It's adapted here. It assesses the reliability and relevance of gene associations by scoring nodes based on how connected they are to the trusted nodes, which in this case is the disease module. So drugs get prioritized if they're close to and strongly connected to that module.
It's like mapping influence in a social network. If a drug targets a protein that has a high trust rank within that disease module, it's highly influential in disrupting the whole malicious signaling process.
Exactly. And closeness centrality complements this by focusing on efficiency. It prioritizes drugs based on the length of their shortest paths to all the other nodes in the network. A drug with high closeness centrality can, in theory, propagate its effect across the network faster.
Okay, so the NEDREX agent takes all this complexity, runs these algorithms, and spits out a ranked list of promising drug candidates.
Complete with a visualization via a tool called drugs.one.
That's a huge amount of computation happening behind the scenes, all from a single chat prompt. But now we have the candidates and the module. How do we make sure that disease module is biologically meaningful and not just some statistical artifact?
Yeah.
We need that same rigor we saw with the entropy validation in the first paper.
Yes, and that is a crucial quality control step. It's handled by the fourth agent, the digest agent. This performs the in silico validation, or what's called functional coherence evaluation.
So it checks if the proteins in that module actually work together functionally.
It does, but it's more rigorous than just listing enriched terms, saying, yes, we found some gene ontology terms related to inflammation.
Right.
Instead of simple enrichment, Digest actually quantifies the coherence of the functional annotations, so gene ontology and KIG-shaped pathways within the module. And then it derives an empirical p-value.
Oh.
By comparing that coherence score to thousands of comparable but randomly selected gene sets of the same size.
So that's a much tougher statistical test. It proves that the functional grouping they found is highly cohesive and significant in a non-random, biologically meaningful way. It ensures they aren't just chasing noise.
It ensures the module truly represents a functional unit of the disease.
Okay, so the data is analyzed, it's validated, it's ranked. Now the system has to put all these findings into scientific context. And that's the job of the research agent.
Yes, this is where the literature grounding comes in, leveraging the vast resources of Semantic Scholar.
And how does this agent use an LLM to perform a better literature search than a human expert might?
It uses task decomposition for precision. Instead of just feeding the initial user query into Semantic Scholar, the LLM-based research agent first parses that inquiry into up to 3 distinct, highly targeted subqueries.
So instead of just searching find papers about resveratrol for Huntington's disease, it might search for 1, resveratrol mechanism of action, 2, resveratrol and Huntington's disease pathway, and 3, clinical trials of resveratrol.
Precisely. This decomposition strategy maximizes the breadth and relevance of the retrieved results. It ensures a comprehensive literature base is built around the predicted therapeutic outcome, and it prevents the system from grounding its answer in just a single, maybe biased source.
Okay, finally, we need the results presented in a format a researcher can actually trust and use. This must be the finalize agent.
The finalize agent is the editor and publisher. It synthesizes all the fragmented outputs from the previous 5 agents, the KG results, the Diamond module, the trust rank rankings, the digest stats, the Semantic Scholar literature.
All of them.
And weaves it into a coherent academic quality answer, usually in Markdown format, complete with citations and descriptions of the methodology.
And this is where the final critical safeguard against LLM fabrication comes in, right?
Yeah.
The output guardrail.
Yes. The finalize agent is itself subject to scrutiny. It employs a separate LLM that is dedicated solely to hallucination verification. This guardrail reviews the streamed response paragraph by paragraph, ensuring it's consistent with the context that was derived directly from the specialized tools.
So if the finalize agent tries to invent a mechanism or misquote a p-value,
The guardrail stops it.
so to summarize the checks and balances, the planning agent ensures the right tools are used in the right order. The KG agent ensures accurate data access with verified IDs. The NEDREX and Digest agents ensure scientific rigor. And the finalize agent and its guardrail ensure the output is factually sound and contextualized. It's an incredibly layered system.
And we can watch this whole flow seamlessly in action using the Huntington's disease use case they present in the paper. A complex analysis becomes a 5-minute conversation.
The user asks which genes are associated with Huntington's.
And the KG agent translates the query, retrieves the seed genes like HTT, GRIK2, and so on.
Then the user asks to run a functional analysis. That triggers the planning agent to engage Diamond to build the molecular module, which is visualized interactively. At the same time, the Digest agent is running that statistical coherence evaluation to confirm the module is biologically robust.
Then comes the critical question. Which existing drugs can target that module? Trust rank and closeness centrality jump into action, identifying candidates like tominersen or resveratrol.
And finally, the user can ask the research agent to find scientific articles about those drugs, which grounds the entire prediction in existing literature via Semantic Scholar.
It's the full translational research pipeline, executed conversationally in real time.
The stark contrast between the internal processes, trust rank cipher, the RPK mupper plus log2 optimization from the prior work, and the simplicity of the user interface, which is just asking a question. That perfectly captures that democratization that was promised years ago.
Which brings us to the most conceptual and I think maybe the most interesting part of this whole deep dive.
The strategic citation.
The strategic citation. Why did the Chatbots paper, with its incredibly sophisticated LLM architecture and multi-agent system, cite that much older 2021 Galaxy and mean stack workflow?
Right. We established they didn't reuse the methodology. They weren't using the RPK upper plus log 2 normalization. They weren't using the Galaxy platform. It wasn't a methodological dependency. So why is it there?
This is where we have to look beyond just the methods and into the rhetorical purpose of academic research. The source material, including notes from one of the authors of that original 2021 paper, confirms this. The citation served a strategic rhetorical function.
Okay.
The citation was used to position Chatbots not as some novel, unvalidated concept of accessibility, but as a natural progression in an established, validated lineage of research.
So the Chatbots authors weren't introducing a brand new problem. They were introducing a revolutionary solution to a recognized and long-documented scientific need.
Exactly. They needed to justify why they built this complex agent system. They needed to show that the difficulty of using bioinformatics tools, the fact that it was expert-only, was a known, documented, and pressing constraint in the biomedical community.
And the Pyers paper served as direct evidence of that. It proved that prior, high-quality translational work had already recognized this pressing need for more accessible and user-friendly interfaces.
It makes their argument so much stronger. It says, look, we tried packaging complexity into web forms and static interfaces with the mean stack, and while it validated the concept, the user experience was still a hurdle. Now, with LLMs, we can finally achieve the full democratization that research 5 years ago already proved was necessary.
It's conceptual borrowing at its finest.
It is. The workflow for rational optimization of cancer chemotherapy, established through that rigorous Shannon entropy validation, is now a conceptual building block for AI drug discovery. It provided the evidence base that computational infrastructure should serve the entire biomedical community, not just programmers.
That is a powerful and very quiet form of academic impact. Your work shifts from being a methodology for one specific cancer type to being the intellectual justification for a completely adjacent next-generation technology.
You proved the need for accessibility, and a later group built the ultimate tool to satisfy that need.
So the value of that foundational work, the RPK upper plus log 2 optimization, the entropy validation, it lives on, not because the code is still running somewhere.
Right.
But because it established the standard for combining scientific rigor with accessibility requirements. It paved the way for the idea that sophisticated drug discovery could, and really should, be conversational.
And the fact that the original author of that foundational paper recognizes this strategic use of their work, that the core philosophy became a conceptual cornerstone, it just highlights the shared scientific value across different generations of technology. The idea of democratization proved its worth, regardless of whether the stack was mean or multi-agent.
What a journey. We've really witnessed the evolution of biomedical computing accessibility. We started with the meticulous validation of complex network science, using entropy metrics, ensuring a data pipeline was calibrated to actual patient survival, and packaging all that complexity into a user-friendly, mean-stack web portal for personalized cancer therapy.
And we arrived at the current state, the creation of this multi-agent AI copilot, Chatbots, which executes entire multi-tool research workflows, from knowledge graph querying using Cypher and embedding-based search to module detection with Diamond and statistical validation with Digest.
All driven by nothing more than a conversational prompt. The focus has decisively shifted from needing to know the code to needing to know the right questions to ask.
The black box of computation is getting significantly friendlier, but the complexity inside, the trust rank algorithms, the entropy calculations, the rigor, that's only increasing.
And that leads us to the final provocative thought we want to leave you with today.
The success of these systems, whether it's the Galaxy pipeline maximizing its correlation to survival data or the Chatbots agents using trust rank, it relies entirely on their ability to translate real-world biological complexity into quantifiable, actionable knowledge.
And as LLMs take on the role of these incredibly effective scientific assistants, conversationally executing these complex multi-step workflows, the crucial culture shifts dramatically.
It's no longer can the machine perform the analysis. Chatbots clearly demonstrates that it can. The question now becomes, can the non-specialist user critically interpret the results and implications that are provided by that black box?
Because automation empowers the user, but the user has to maintain that critical thinking to avoid just blindly trusting the output.
Exactly. The future of personalized medicine will be defined by that constant balance. The AI provides the answer. The human must provide the context and the clinical judgment.
That's a powerful message for researchers and clinicians alike. And if you, the listener, are curious to dive deeper into the mathematical and biological concepts that underpin this whole evolution, the network science, the entropy metrics, the logical thinking that drives these models. I highly recommend exploring the specialized material on innovating with biomathematics that form the basis of the foundational work we discussed today. We'll make sure to provide a link for those resources in the description for anyone interested in seeing how these ideas are built from the ground up.
Thank you for joining us on this deep dive into the accessibility lineage of biomedical computing.
We'll catch you next time for more complexity explained simply.